Hello and welcome, it's Dr. Fred Frizzle here, and today I'm going to be continuing my series on GCSE OCR specification biology. Right, okay, so first of all I've brought you up here the chemical symbol equation for photosynthesis. So it's the conversion of carbon dioxide and water into oxygen and glucose. Okay, so the next thing I want to go through is the idea of reactants and products. Now you might be familiar with this from chemistry mostly, because this is obviously a chemical equation which is used in biology. Okay, so reactants, are the things that react in order to form these products. So these are converted into the products which are over this side of the equation. Now, how does this conversion take place? Well, first of all, there are a number of different things that a plant needs in order to photosynthesize, one of which is water, one of which is carbon dioxide, one of which is light from the sun. And this light energy hits these palisade mesophyll cells. Now, if you watched my last video, you'll know where the palisade mesophyll cells are located. So I suggest you watch my last video on this particular playlist, because that will show you the anatomy of the leaf and exactly where in the leaf this photosynthesis takes place in the palisade mesophyll cells. And this is because these cells contain a high number of chloroplasts. Now you don't need to know for this level of biology exactly sort of where in the chloroplast photosynthesis actually takes place by the light hitting the chlorophyll inside it. You don't need to know exactly where the chlorophyll is. But anyway, the chlorophyll is the green pigment in plants which makes leaves green basically, and that is where the light hits in order to initiate this process of photosynthesis. Okay, so once photosynthesis has occurred and it's generated these particular products, then what can happen is the next process called respiration. I say the next, it doesn't have to happen directly afterwards, but because these are almost like reciprocal reactions, which basically means like one of them occurs and then the next one can occur from the products of it. So essentially this creates oxygen and glucose, then that oxygen and glucose can be reused in respiration to be converted back into carbon dioxide and water, which this needs to start again. So it's almost like a cycle that takes place within animals and plants, because obviously plants respire as well as photosynthesize because they need to get energy for growth and things. And the next thing I want to talk to you a little bit about is enzymes. Now, enzymes you might think aren't linked to photosynthesis. Well, actually they are, because there's a number of different enzymes that you don't need to worry about at this stage in your education. But there are a number of different enzymes that are associated with photosynthesis, and as the process progresses and you learn more detail about the individual stages, and I've actually made some videos on this, you'll learn that there are particular enzymes that are involved. Okay, so what an enzyme does is it breaks down a molecule known as a substrate into products. So there's lots of different types of enzymes. I wrote down for you one here, which is lipase, which converts fats into glycerol fatty acids. There are loads of different types of enzymes, but you don't really need to know about too many. The key ones I think are lipase, protease, and amylase. You should really know what they all do. So lipase, as I've just said, breaks down fats into glycerol and fatty acids. Then you have amylase that breaks down starch into simple sugars such as glucose. And then you have things like protease that breaks down your protein into something called amino acids. Now these amino acids that protein can be broken down into, like how do those amino acids come together to form new proteins? Well, we need genes to code for these new proteins, so that's basically what genes do. They basically contain instructions which tell the cell in what order to put together amino acids into a chain in order to form a protein. Right, okay, so enzymes don't always work. They only work like around this temperature of 37 degrees, which is called their optimum temperature. The same thing happens with pH. I don't know if you're familiar with the pH scale, but basically there's a scale between strong acid and strong alkali, 0 being strong acid and 14 being strong alkali. And basically 37 degrees and about, it varies per enzyme. I can't really tell you exactly where on the pH scale enzymes work best, but all you need to know is this end is acid and this end is alkali, and some enzymes work best lower down the scale and some work best higher up the scale. And the same goes for temperature. I mean, all enzymes work at different temperatures, but supposing the temperature is too high for a particular enzyme, what that will result in is the enzyme becoming denatured. Also, equally, what can happen if the temperature is too low is, as you can see here, I've drawn you an enzyme with its active site here, which is where the substrate molecule fits into it. So what happens is, supposing that substrate doesn't collide with it with enough energy, because it doesn't have enough energy, like due to not having enough temperature, because temperature, don't forget, as you increase temperature you also increase energy. If the collision doesn't have enough energy, then this reaction of actually the breakdown of this, or metabolism if you like, of this substrate won't actually take place. So that's quite an important concept to get your head around. Also, the other thing is these substrates can be built up from smaller molecules, and there's a particular type of enzyme that does this, that takes part in anabolic reactions. So it's an anabolic reaction enzyme that's used for this, and what this does is it builds up like simple sugars into things like starches, which can be stored in plants. And basically plants want to store starch and not glucose, because glucose can bloat other cells by diffusion in between the cells. So basically that's good for compact storage in plants. So I hope this video has been helpful, and hope it's given you a general overview. The one other thing is the lock and key method. That just basically means that this substrate has to fit perfectly into this active site of the enzyme. I'll go into in more detail with a few different theories on how these particular substrates fit into the active site of enzymes, because there are a few other theories. We don't need to worry too much about those. So I'll see you in the next video. Thanks for watching.